Chapter 13. How Cells Obtain Energy from Food. As we discussed in Chapter 3, cells require a constant supply of energy to generate and maintain the biological order that keeps them alive. This energy comes from the chemical bond energy in food molecules, which thereby serve as fuel for cells. Perhaps the most important fuel molecules are the sugars. Plants make their own sugars from CO2 by photosynthesis. Animals obtain sugars and other molecules, such as starch, that are easily broken down into sugars by eating other organisms. Nevertheless, the process whereby these sugars are oxidized to generate energy is very similar in both animals and plants. In both cases, the cells that form the organism harvest useful energy from the chemical bond energy locked in sugars as the sugar molecule is broken down and oxidized to carbon dioxide, CO2, and water, H2O. This energy is stored as high-energy chemical bonds, covalent bonds that release large amounts of energy when hydrolyzed, in activated carrier molecules such as ATP and NADPH. These carrier molecules, in turn, serve as portable sources of the chemical groups and electrons needed for biosynthesis, discussed in Chapter 3. In this chapter, we trace the major steps in the breakdown or catabolism of sugars and show how this oxidation produces ATP, NADH, and other activated carrier molecules in cells. We concentrate on the breakdown of glucose because these reactions dominate energy production in most animal cells. A very similar pathway operates in plants, fungi, and many bacteria. Other molecules, such as fatty acids and proteins, can also serve as energy sources if they are funneled through appropriate enzymatic pathways. We also see how many of the molecules generated from the breakdown of sugars and fats can be used to build the macromolecules in cells. Finally, we examine how cells regulate their metabolism and how they store food molecules for their future metabolic needs. We will save our discussion of how cells produce most of the ATP that they need for Chapter 14. The breakdown and utilization of sugars and fats. If a fuel molecule such as glucose were oxidized to CO2 plus and H2O in a single step, as happens in non-living systems, it would release an amount of energy many times larger than any carrier molecule could capture. Instead, living cells use enzymes to carry out the oxidation of sugars in a tightly controlled series of reactions. As illustrated in Figure 13.1, a glucose molecule is degraded step by step, paying out energy in small packets to activated carrier molecules by means of coupled reactions. In this way, much of the energy released by oxidizing glucose is saved in the high-energy bonds of ATP and other activated carrier molecules and made available to do useful work for the cell. Animal cells make ATP in two ways. First, certain steps in a series of enzyme-catalyzed reactions are directly coupled to the energetically unfavorable reaction, ADP plus P subscript I to ATP. The oxidation of food molecules provides the energy that allows this unfavorable reaction to proceed. Most ATP synthesis, however, takes place in mitochondria and uses the energy from activated carrier molecules to drive ATP production. This process involves the mitochondrial membrane and will be described in detail in Chapter 14. In this chapter, we focus on the first sequence of reactions by which food molecules are oxidized, both in the cytosol and inside the mitochondria. These reactions produce both ATP and the activated carrier molecules that will subsequently drive the production of much larger amounts of ATP in the mitochondrial membrane. Food molecules are broken down in 3 stages. The proteins, lipids, and polysaccharides that make up most of the food we eat must be broken down into smaller molecules before our cells can use them, either as a source of energy or as building blocks for other molecules. This breakdown process, which uses enzymes to degrade complex molecules into simpler ones, is dubbed catabolism. Catabolism must act on food taken in from outside, but not on the macromolecules inside our own cells. Therefore, stage 1 in the enzymatic breakdown of food molecules, digestion, occurs either outside cells in our intestine or in a specialized organelle within cells called the lysosome. A membrane that surrounds the lysosome keeps its digestive enzymes separated from the cytosol, discussed in Chapter 15. Digestive enzymes reduce the large polymeric molecules in food into their monomeric subunits, proteins in amino acids, polysaccharides into sugars, and fats into fatty acids and glycerol. After digestion, the small organic molecules derived from food enter the cytosol of a cell, where their gradual oxidation begins. As illustrated in Figure 13.2, this oxidation occurs in 2 further stages. Stage 2 starts in the cytosol and ends in mitochondria, while stage 3 is confined to the mitochondria. In stage 2 of cellular catabolism, a chain of reactions called glycolysis converts each molecule of glucose into 2 smaller molecules of pyruvate. Sugars other than glucose can also be used after being first converted to one of the sugar intermediates in this glycolytic pathway. During the formation of pyruvate, 2 types of activated carrier molecules are produced, ATP and NADH. The pyruvate is then transported from the cytosol into the mitochondrion's large internal compartment, or matrix. There, a giant enzyme complex converts each pyruvate molecule into CO2 plus acetyl-CoA, another of the activated carrier molecules discussed in Chapter 3. See Figure 336. Large amounts of acetyl-CoA are also produced by the stepwise breakdown and oxidation of fatty acids derived from fats. Stage 3 of the oxidative breakdown of food molecules takes place entirely in mitochondria. The acetyl group in acetyl-CoA is transferred to a molecule called oxaloacetate to form citrate, which enters a series of reactions called the citric acid cycle. The transferred acetyl group is oxidized to CO2 in these reactions, and large amounts of high-energy electron carrier NADH are generated. Finally, the high-energy electrons from NADH are passed along a series of enzymes within the mitochondrial inner membrane, called an electron transport chain, where the energy released by their transfer is used to drive a process that produces ATP and consumes molecular oxygen, O2 gas. It is in these final steps that most of the energy released by oxidation is harnessed to produce most of the cell's ATP. Through the production of ATP, the energy derived from the breakdown of sugars and fats is redistributed as packets of chemical energy in a form convenient for use in the cell. Roughly 10 to the 9 molecules of ATP are in solution in a typical cell at any instant, and in many cells, all of this ATP is turned over, that is, used up and replaced every 1 to 2 minutes. An average person at rest will hydrolyze his or her weight in ATP molecules every 24 hours. In total, nearly half of the energy that could, in theory, be derived from the oxidation of glucose or fatty acids to H2O and CO2 is captured and used to drive the energetically unfavorable reaction P subscript I plus ADP to ATP. By contrast, a modern combustion engine, such as a car engine, can convert no more than 20% of the available energy in its fuel into useful work. In both cases, the remaining energy is released as heat, which in animals helps to make our bodies warm. Glycolysis is a central ATP-producing pathway. The central process in stage 2 of the breakdown of food molecules is the degradation of glucose in the sequence of reactions known as glycolysis. From the Greek glyx, sweet, and lysis, splitting, glycolysis produces ATP without the involvement of O2. It occurs in the cytosol of most cells, including many anaerobic microorganisms. Glycolysis probably evolved early in the history of life, before photosynthetic organisms introduced oxygen into the atmosphere. During glycolysis, a glucose molecule which has 6 carbon atoms is cleaved into 2 molecules of pyruvate, each of which contains 3 carbon atoms. For each molecule of glucose, 2 molecules of ATP are consumed to provide energy to drive the early steps, but 4 molecules of ATP are produced in the later steps. Thus, at the end of glycolysis, there is a net gain of 2 molecules of ATP for each glucose molecule broken down. Glycolysis involves a sequence of 10 separate reactions, each producing a different sugar intermediate, and each catalyzed by a different enzyme. These reactions are presented in outline in Figure 13.3 and in detail in Panel 13.1, pages 430 to 431. Like most enzymes discussed in Chapter 4, the enzymes that catalyze glycolysis all have names ending in ase, like isomerase and dehydrogenase, which specify the type of reaction they catalyze. Although no molecular oxygen is involved in glycolysis, oxidation occurs. Electrons are removed from some of the carbons derived from glucose by NAD+, producing NADH. The stepwise nature of the process allows the energy of oxidation to be released in small packets, so that much of it can be stored in carrier molecules rather than all of it being released as heat. See Figure 13.1. Some of the energy released by this oxidation drives the synthesis of ATP molecules from ADP and P subscript I. The synthesis of ATP in glycolysis is known as substrate level phosphorylation because it occurs by the transfer of a phosphate group directly from a substrate molecule, a sugar intermediate, to ADP. The remainder of the energy harnessed during glycolysis is stored in the electrons in NADH. 2 molecules of NADH are formed per molecule of glucose in the course of glycolysis. In aerobic organisms, these NADH molecules donate their electrons to the electron transport chain, as described in detail in Chapter 14. The electrons are passed along this chain to O2, forming water, and the NAD+ formed from the NADH is used again for glycolysis. See Step 6 in Panel 13.1, page 431. The piecing together of the complete glycolytic pathway in the 1930s was a major triumph of biochemistry, and it was quickly followed by the recognition of the central role of ATP in cellular processes. Fermentations allow ATP to be produced in the absence of oxygen. For most animal and plant cells, glycolysis is only a prelude to the third and final stage of the breakdown of food molecules, in which large amounts of ATP are generated by oxidative phosphorylation in mitochondria with the consumption of oxygen. However, for many anaerobic microorganisms which do not use O2 and can grow and divide in its absence, glycolysis is the principal source of ATP. The same is true in certain animal tissues, such as skeletal muscle, that can continue to function at low levels of O2. In these anaerobic conditions, the pyruvate and the NADH stay in the cytosol. The pyruvate is converted into products that are excreted from the cell. For example, lactate in muscle or ethanol and CO2 in the yeasts used in brewing and bread making. In the process, the NADH gives up its electrons and is converted back into NAD+. This regeneration of NAD+ is required to maintain the reactions of glycolysis. Figure 13.4. Anaerobic energy-yielding pathways like these are called fermentations. Studies of the commercially important fermentations carried out by yeasts inspired much of early biochemistry. Many bacteria and archaea can also generate ATP in the absence of oxygen by anaerobic respiration, a process that uses a molecule other than oxygen as a final electron acceptor. Anaerobic respiration differs from fermentation in that it involves an electron transport chain embedded in a membrane. Glycolysis illustrates how enzymes couple oxidation to energy storage. The paddle wheel analogy in Chapter 3 explained how cells harvest useful energy from the oxidation of organic molecules by coupling an energetically unfavorable reaction to an energetically favorable one. See Figure 330. To illustrate exactly how enzymes, the paddle wheel in our analogy, allow coupled reactions to occur, we take a closer look at a key pair of glycolytic reactions. These reactions, steps 6 and 7 in panel 13.1, convert the 3-carbon sugar intermediate glyceraldehyde 3-phosphate, an aldehyde, into 3-phosphoglycerate, a carboxylic acid. This conversion entails the oxidation of an aldehyde group to a carboxylic acid group, which occurs in 2 steps. The overall reaction releases enough free energy to convert a molecule of ADP to ATP and to transfer 2 electrons from the aldehyde to NAD+ to form NADH, while still releasing enough heat to the environment to make the overall reaction energetically favorable. The delta G degrees for the overall reaction is negative 3.0 kilocalories per mole, negative 12.5 kilojoules per mole. This remarkable feat of energy harvesting is detailed in Figure 13.5. The chemical reactions are precisely guided by 2 enzymes to which the sugar intermediates are tightly bound. In fact, as shown in Figure 13.5, the first enzyme, glyceraldehyde 3-phosphate dehydrogenase, forms a short-lived covalent bond to the aldehyde through a reactive SH group on the enzyme and catalyzes its oxidation in this attached state. The reactive enzyme-substrate bond is then displaced by an inorganic phosphate ion to produce a high-energy phosphate intermediate, which is released from the enzyme. The intermediate, 1,3-bisphosphoglycerate, binds to the correct enzyme, phosphoglycerate kinase. This enzyme then catalyzes the energetically favorable transfer of the intermediate's high-energy phosphate to ADP, forming ATP and completing the process of oxidizing an aldehyde to a carboxylic acid. We have discussed this particular oxidation process in some detail because it provides a clear example of enzyme-mediated energy storage through coupled reactions. Figure 13.6. These reactions, step 6 and 7, are the only ones in glycolysis that create a high-energy phosphate linkage directly from inorganic phosphate. As such, they account for the net yield of 2 ATP molecules and 2 NADH molecules per molecule of glucose. As we have mentioned, this NADH must be reoxidized to the NAD+ required for these coupled reactions. If NAD+ is not available, glycolysis will stop. See Figure 13.4. As we have just seen, ATP can be readily generated from ADP when reaction intermediates are formed with phosphate bonds of higher energy than those in ATP. The energy of phosphate bonds can be ordered by determining the standard free energy change, delta G degrees, for the breakage of each bond by hydrolysis. Figure 13.7 compares the high-energy phosphoanhydride bonds in ATP with some other phosphate bonds that are generated during glycolysis. As explained in panel 3.1, page 95, we describe bonds as high energy only in the sense that their hydrolysis is particularly energetically favorable. Sugars and fats are both degraded to acetyl-CoA in mitochondria. In aerobic metabolism in eukaryotic cells, the pyruvate produced by glycolysis is actively pumped into the mitochondrial matrix, the major internal compartment of this organelle. See Figure 14.12. There, it is rapidly decarboxylated by a giant complex of 3 enzymes called the pyruvate dehydrogenase complex. The products of pyruvate decarboxylation are a molecule of CO2, a waste product, a molecule of NADH, and a molecule of acetyl-CoA. The structure and function of pyruvate dehydrogenase are outlined in Figure 13.8. Fatty acids derived from fat are an alternative fuel to sugars for energy generation. Like the pyruvate derived from glycolysis, fatty acids are converted into acetyl-CoA in mitochondria. Each long molecule of fatty acid in the form of the activated molecule fatty acyl-CoA is broken down completely by a cycle of reactions that trims 2 carbons at a time from its carboxyl end, generating 1 molecule of acetyl-CoA for each turn of the cycle. A molecule of NADH and a molecule of another electron carrier, FADH2, are also produced in this process. Figure 13.9. Sugars and fats provide the major energy sources for most non-photosynthetic organisms, including humans. In the course of their processing to acetyl-CoA, only a small part of the useful energy stored in these foodstuffs is extracted and converted into ATP or NADH. Most of the energy is still locked up in acetyl-CoA. The next stage in respiration, in which the acetyl group in acetyl-CoA is oxidized to CO2 and H2O in the citric acid cycle, is therefore central to the energy metabolism of aerobic organisms. In eukaryotes, the citric acid cycle takes place in mitochondria, the organelles to which pyruvate and fatty acids are directed for acetyl-CoA production. Figure 13.10. In addition to pyruvate and fatty acids, some amino acids are transported from the cytosol into mitochondria, where they are also converted into acetyl-CoA or one of the other intermediates of the citric acid cycle. See Figure 13.2. Thus, in the eukaryotic cell, the mitochondrion is the center towards which all energy-yielding processes lead, whether they begin with sugars, fats, or proteins. In aerobic bacteria, which have no mitochondria, all of these reactions, glycolysis, acetyl-CoA production, and the citric acid cycle, take place in the single compartment of the cytosol. The citric acid cycle generates NADH by oxidizing acetyl groups to CO2. The third and final stage in the oxidative breakdown of food molecules to generate energy requires abundant O2. Although living organisms have inhabited Earth for the past 3.5 billion years, the planet is thought to have developed an atmosphere containing O2 gas only some 1 to 2 billion years ago. See Figure 1443. The oxygen-consuming reactions we discuss next are therefore likely to be of relatively recent origin. In contrast, the mechanism used to produce ATP in Figure 13.5 does not require oxygen, and relatives of that elegant pair of coupled reactions could have arisen very early in the history of life on Earth. In the 19th century, biologists noticed that in the absence of air, anaerobic conditions, cells produce lactic acid, for example in muscle, or ethanol, for example in yeast, while in the presence of air, aerobic conditions, these cells consume O2 and produce CO2 and H2O. Intensive efforts to define the pathways of aerobic metabolism eventually focused on the oxidation of pyruvate and led in 1937 to the discovery of the citric acid cycle, also known as the tricarboxylic acid cycle, or the Krebs cycle. See How We Know, pages 440 to 441. The citric acid cycle accounts for about 2/3 of the total oxidation of carbon compounds in most cells, and its major end products are CO2 and high-energy electrons in the form of NADH. The CO2 is released as a waste product, while the high-energy electrons from NADH are passed to a series of membrane-bound enzymes known collectively as the electron transport chain. At the end of the chain, these electrons combine with O2 to produce H2O. The citric acid cycle itself does not use O2. However, it requires O2 to proceed because the electron transport chain allows NADH to get rid of its electrons and thus regenerate the NAD+ that is needed to keep the cycle going. The citric acid cycle, which takes place in the mitochondrial matrix, catalyzes the complete oxidation of the carbon atoms of the acetyl groups in acetyl-CoA, converting them into CO2. The acetyl group is not oxidized directly, however. Instead, it is transferred from acetyl-CoA to a larger 4-carbon molecule, oxaloacetate, to form the 6-carbon tricarboxylic acid, citric acid, for which the subsequent cycle of reactions is named. The citric acid molecule is then gradually oxidized, and the energy of this oxidation is harnessed to produce energy-rich carrier molecules, in much the same manner as we described for glycolysis. The chain of 8 reactions forms a cycle, because the oxaloacetate that began the process is regenerated at the end, as shown in outline in Figure 1311. We have so far discussed only one of the 3 types of activated carrier molecules that are produced by the citric acid cycle, NADH. In addition to 3 molecules of NADH, each turn of the cycle also produces 1 molecule of FADH2, reduced flavin adenine dinucleotide, from FAD and one molecule of the ribonucleotide GTP, guanosine triphosphate, from GDP. See Figure 1311. The structures of these two activated carrier molecules are illustrated in Figure 1312. GTP is a close relative of ATP, and the transfer of its terminal phosphate group to ADP produces one ATP molecule in each cycle. Like NADH, FADH2 is a carrier of high-energy electrons and hydrogen. As we discuss shortly, the energy that is stored in the readily transferred high-energy electrons of NADH and FADH2 are subsequently used to produce ATP through the process of oxidative phosphorylation, which occurs in the mitochondrial membrane. Oxidative phosphorylation is the only step in the oxidative catabolism of foodstuffs that directly requires O2 from the atmosphere. The complete citric acid cycle is presented in panel 13.2, pages 442 to 443. Note that the oxygen atoms required to make CO2 from the acetyl groups entering the citric acid cycle are supplied not by O2 but by water. As illustrated in the panel, 3 molecules of water are split in each cycle, and the oxygen atoms of some of them are ultimately used to make CO2. A common misconception about aerobic respiration is that the atmospheric O2 required for the process is converted into the CO2 that is released as a waste product. In fact, as we shall see, the molecules of O2 gas are reduced to water, not incorporated directly into CO2. Many biosynthetic pathways begin with glycolysis or the citric acid cycle. Catabolic reactions, such as those of glycolysis and the citric acid cycle, produce both energy for the cell and the building blocks from which many other molecules are made. See Figure 3.2. So far, we have emphasized energy production rather than the provision of starting materials for biosynthesis. But many of the intermediates formed in glycolysis and the citric acid cycle are siphoned off by biosynthetic or anabolic pathways, where they are converted by a series of enzyme-catalyzed reactions into amino acids, nucleotides, lipids, and other small organic molecules that the cell needs. Oxaloacetate and alpha-ketoglutarate from the citric acid cycle, for example, are transferred from the mitochondrion back to the cytosol, where they serve as precursors for many essential molecules, such as the amino acids aspartate and glutamate, respectively. An idea of the complexity of this process can be gathered from Figure 1317, which illustrates some of the branches leading from the central catabolic reactions to biosyntheses. Electron transport drives the synthesis of the majority of the ATP in most cells. We now return to the last stage in the oxidation of a food molecule, the stage in which most of its chemical energy is released. In this final process, the electron carriers NADH and FADH2 transfer the electrons they have gained by oxidizing other molecules to the electron transport chain. This specialized chain of electron carriers is embedded in the inner membrane of the mitochondrion in eukaryotic cells, in the plasma membrane of bacteria. As the electrons pass through the series of electron acceptor and donor molecules that form the chain, they fall to successively lower energy states.